Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And a very good day to Dr. Alina and Dr. Nazaria. So inshallah for today, my name is Muhammad Zahid Bezu Akli with metric number 1170313. So for today, I will proposing my proposal for my FYP final year project on title Proximate Analysis of Freeze-dried Ipomoea Batatas leaf sweet potato leaf juice. Basically, the Ipomoea Batatas leaf is the scientific name for the sweet potato leaf. Okay, so as you can see, so this slide is prepared by me, myself, and I have been supervised with Dr. Shamila Binti Mansur. So, without hesitating, let's bring it on. Okay, so basically for this proposal defense session, I will tell in 3 chapters. I will present my proposal in 3 chapters, which is for the chapter 1 is introduction. So for the introduction, as we can see, I just put one figure and one table. So for the figure 1, which is the sweet potato leaves itself, and the table 1, which is nutrient composition for the sweet potato leaf. Okay, basically, as we know, all the plants, especially for the green plant, has basically for all plants and for all green plants especially have their own nutrient inside them. So it same goes to the sweet potato leaf also. Okay, so as we can see for the table 1 in nutrient composition, okay, I had this table from Jabatan Pertanian Negeri Pulau Pinang. So this table show about 15 nutrient that contain in sweet potato leaf. Okay, then so for the research problem of my proposal, which is what's made me to do this project. Okay, basically, here I just put the one figure. Okay, so this figure shows about the highest hectareage for 10 types of vegetables and cash crops for the year 2018. Okay, so this table, this statistic is come out from the Jabatan Pertanian Semenanjung Malaysia in their statistik tanaman sayur-sayuran and tanaman ladang 2018. So in this pie chart, we can see that the sweet potato is in the 9th place of the highest hectares for 10 types of vegetable. So can we imagine that if the production of the sweet potato in 2018 means that the production of the sweet potato in Malaysia is higher. So can we imagine how the kilo of tons of the waste from the sweet potato, which is the leaf itself, will be wasting like that? So instead of we just wasting the sweet potato leaf, why not we come out with some project, or we come out with some works to analyze a little bit a nutrient content in the sweet potato leaf. Okay, so for the research question and the RO research objective for this proposal, for my proposal is for the research question, the first is how many nutrients are left over in a different period of time. And the second is what is the minimum amount of nutrient content left after pasteurization process. And the third one is pasteurized and non-pasteurized samples are different. Okay, and for the research objective, we have what I want to know is I want to analyze the moisture content, protein content, and fat content of sweet potato leaf in the 3 different period of time, which I will investigate in week 1, week 3, and week 5. And to figure out the effect of freeze-dried process on the antioxidant activity of sweet potato leaf, and also to determine the most stable between pasteurized sample or non-pasteurized sample. Okay then, so for the chapter 2 in the literature review session, I just come out with two literature review, which is about the proximate analysis and the effect of pasteurization. So as we know, the proximate analysis is a quantitative analysis of food to determine the percentage of fat, fiber, protein, carbohydrate, moisture, and ash, which is this state from the Merriam-Webster dictionary. And actually, for the proximate analysis, it has about 8 categories, which is moisture content, protein, fat, available, carbohydrate, total ash, total dietary fiber, total sugar, and energy. Okay, so I just I just focus on not all the proximate analysis, but I just focus on some of the proximate analysis, the category of the proximate analysis. So for the effect of pasteurization, as we know, if we pasteurize, the shelf life of the food will be more than the food that have the non-pasteurized. So that's we know about that. Okay, so for the chapter 3 research methodology, just I gave a simple one, which is the flowchart of my work for the next sem. Okay, so the first one, I will get the samples and then I will blend it and I will filter and get the juice, and from the juice I will make the juice to go through with the freeze dry process, and then I will divide to pasteurized and non-pasteurized, and then for the sample I will do the proximate analysis of the sample and collect the data and then do a report.